Hello.
I apologize for the late start. We're having some network issues. They're doing some work around my place. So let's see how it goes. Yeah. By the way, anyone has any questions while we wait for others to join?
We have our first exam next Saturday, correct?
No, that is a schedule that we have and it's tentative to us. I don't think it's going to be next Saturday because we need to review the material. I would like for us to cover the material that we have today. And then I will give you a study guide. And then when we meet next week, you will tell me, we will kind of go over the review and everybody will tell me their comfort level of the material or if you have any questions. Then until then after that, we could decide to do the test. Does that make sense?
Yeah, thank you. Because I was going to ask, are we going to get a study guide or anything? But yeah, you answered all my questions. Thank you.
Any other question?
But we do have a quiz today, right?
We do have a quiz today. Every week we have a quiz. And again, as I had indicated, the purpose of these quizzes is more like a review opportunity to keep you so that the material doesn't build up too much. And it allows you to be able to review your material. And then when, like we say, we're going to have a midterm, it's not going to be something too different because it's just going to be, the format is going to be similar to what you have for the quizzes. And then what you will be doing now is just doing a longer version of the quiz, if that makes sense. Does that make sense?
Yes. Are we going to be taking the quiz in lab? And do we need to bring anything?
Well, no, for this quiz, the quiz that you're going to have this week is just going to be for the material that I'm going to cover for this week. The exam is a midterm that will be like a summary of 6 chapters that we have done.
Okay, but where do we take the quiz? Are we taking it during the lab or?
No, the quiz is going to be done online.
Okay.
Yeah. Okay. Let me get started so that we don't run into any other issues, and let's get going. So the last time we met, we were talking about cells. And did you guys get a chance to look at the chapter? And I wanted to find out if you had any questions. Let me share my screen here. If we get kicked out, just join back in. I will just wait like 5 minutes. I will join back in case something goes wrong. Just give you a heads up. Can you see my screen? Can you see the screen? Yeah, okay.
Yes.
Excellent. Okay. Let me put in on presentation mode so that we see, push this up all the way. What's my mouse? It's over here. So we went around talking about cells, how possibly cells came about, and then we were asking questions like, is there any benefit of the fact that cells are so super small that they are microscopic, that we could see only using the microscope? And then I presented the fact that there is a benefit to the surface to area area to volume ratio that allows for easy material transport in and out of cells, and that could be a deductive benefit of that. And so, because cells material get into and out of our cells through the entire plasma membrane. And then we went ahead, we looked at, talks about different microscopes, which we tried to use some last week, using the light microscopes last week, trying to examine various things on it, and then learning how to be able to view something under the microscope. We talked about the various kinds of magnifications and that different microscopes have, indicating that the electron microscopes, they are great at having a higher magnification, being able to see very, very small material expanded. However, you cannot see living things using the electron microscope because of the way we need to prepare the sample. Then we did a comparison view. You guys, this is the electron microscope picture of what you were seeing, something round like this. This is like a cross section of a blood vessels, but the cells that you were looking at around 40x, you were seeing round circles like this of red blood cells. Then I talked to you about the differences between prokaryotic and eukaryotic cells, indicating it looks like the first cell type that came about was a prokaryotic cell. Then we had a eukaryotic cell that probably acquired some material into it to form organelles. And the rationale behind that is that things like the mitochondria are almost the size of a bacterial cell. And then in the terms of plants, the chloroplast is almost the size of a bacterial cell. Contrasting these two cell types, we said is the presence of organelles in the eukaryotic cell that is absent in the membrane-bound nucleus in the eukaryotic cell that is absent in the prokaryotic cell. And then we also indicated that in terms of similarities, we see both cell types having nucleic acid, in this case DNA, and then we moved ahead and then we looked at they share plasma membranes, but in addition to the prokaryotic cells, they had sometimes some of them would have like cell walls that eukaryotic cells do not have. Then we look at this prototype of a eukaryotic cell, naming all the parts, talking about understanding what each part does. Then this is the process again that like the original prokaryotic cell that took up material and organelles like this mitochondria mentioned to eventually become the eukaryotic cells. Then we talked about the cell membrane. We looked at its architecture, indicating that it's mainly made up of phospholipids. To be more specific, it's a phospholipid bilayer with the sites that have the phosphates on the exterior part and the interior part of the cell. Meanwhile, the fatty acid chains are within, pointing in the center of the phosphobilipid. And I said the membrane is mainly phospholipids. I didn't say it was only phospholipids. The reason being that we have membrane, transmembrane proteins like these ones. Some of them, this purpose they serve is to form like channels for ionic molecules to be able to move in and out of the cell. And then we have cholesterol molecules that are impregnated here and there to maintain the architecture of the cell. And onto some of these plasma membranes are carbohydrate chains, some of which serve as receptor sites for the cells to be able to identify certain things like surface receptors. And then in terms of how things get into and out of the cell, we say the membrane is semi-permeable. Semi-permeable because it will selectively have certain things get in. Ionic things can only get in through these channels. Meanwhile, things that are non-ionic, polar molecules or hydrophobic molecules would only can diffuse in and out of the membrane. So we said, for example, this is an example of having a transmembrane protein. The water molecule is polar because this red side here, which is like indicating the oxygen molecule, is slightly negative. The white ones are slightly positive. So with that charged nature of the molecule, it cannot diffuse in and out. It would depend on a transmembrane corridor, this transmembrane protein to come through. And in this case, the water molecule, the name of the transmembrane protein that moves water in and out of the cell is called aquaporin. Molecules that are not charged can diffuse in and out. And those molecules are usually the hydrophobic molecules. Hydrophobic. Phobic does not like. Hydro is water. It does not like water. Hydrophobic molecules can diffuse in and out. And large molecules can be brought in through through phagocytosis or endocytosis. So these are basically the mechanisms of transport of material in and out of the membrane. You need to know these ones, the differences between each. Diffusion, simply molecules moving from an area where they are in higher concentration to an area where they are in lower concentration. So if we are talking about the cell, now we are looking if the molecules inside the cell, for example, are more than they are outside the cell, then the molecules will move out of the cell. And reversely, if the molecules on the outside are in higher concentration than the molecules inside, they would diffuse into the cell until they attain a point where they have similar concentrations on the outside and the inside. In that case, they say they have attained equilibrium. And when they attain equilibrium, that doesn't mean that the molecules then stop. All it simply means is that the rate at which the molecules are coming in is equal to the rate at which they are getting out. And then osmosis, we say the simplest way to define is that it is when you are referring to the diffusion of water molecules. If you are talking about water molecules moving in and out of the cell, you refer to that process where they are moving of higher concentration to lower concentration as osmosis. Facilitated transport, facilitate just means like facilitate to make easy. So you could imagine that if you were considering like the water molecules, if you were considering like charged molecules where they need to move through a transmembrane protein, because that corridor within the protein in which they move through, we said that is facilitated transport because it is through that corridor, through that opening, that hole within the transmembrane protein that they are able to move in and out. Active transport, on the other hand, is when molecules are moving against their concentration gradient. Actually, let me push this forward so that we could look at some of the pictures. Diffusion, here the molecules are in equilibrium. Molecules here, we're talking about these red and purple. Here, they are more concentrated this way. Here, they are almost equal number on each side after an equilibrium, osmosis. And then we talked about hypertonic and hypotonic solution. And somebody was generous to see that I indicated something that is wrong. Who can tell me what is wrong with this first bullet point? Can anybody see what is wrong with any of these bullet points? Are you still there? Can you hear me?
Yeah, we can hear you.
Yeah.
Okay. Thank you. Thank you. I was just making sure that I've not been talking to myself. Read these bullet points here and tell me if you could picture, especially the first two, tell me that if you could see anything wrong or there's nothing wrong from your own understanding of the explanation of these two terms, hypertonic and hypotonic. Okay, let's look at this. When we say hypertonic, we say hypertonic solutions have more solid than, there's more solid. That is, so if you just imagine that when you take salt and you are dividing it, you are dissolving in a glass of water. So the salt is the solute, the water is the solvent. So when they mix up, they form a solution. Okay. So inside our cells, our red blood cells, for example, which is what we are looking at here, inside our red blood cells, they have water, and they also have some salts dissolved in it. So when you are talking about these solutions, you are comparing, what if you take red blood cells, which are floating in our blood, and then you look at the blood in which the red blood cells are swimming inside. Our blood too also has salts in it. So if you take red blood cells, for example, and you put them in a solution of a salt solution, like in this case in a beaker, meaning that you have some salt inside the water that is in the beaker, and you have some salt inside the cell that are in the cytoplasm. The question is, which way will water move between the salt in the beaker and the one in the cells? Now, if the concentration of the salt in the water outside is the same as the concentration of the salt inside the cells, that solution is isotonic. I said isotonic solutions have equal amounts of solutes inside and outside of the cell, which is in this case. So the rate at which the water is coming into the cell, it will be the rate at which the water is getting out of the cell. So everything kind of balances out. But what if the concentration of salt in the water outside was the concentration was lower, meaning that there's less salt in this water in terms of concentration and more salt in the cell in terms of concentration? The tendency would be that water will move from outside, since the water molecules are many more, and then they will start trying to flow inside the cell to equilibrate. In this situation where the concentration of salt in the water outside is less, we actually should refer to this as hypotonic solution. So hypotonic solutions have less salt concentration than there is in the cell. So that is where this thing is wrong, because here I should have written hypotonic here, which means that if this situation where the water was there's more water molecules and less salt molecules here, water would go in here and it would swell on and burst the cells. So this should be hypotonic. And then this other one here, which I put here, I say solutions have less solute So this one should be here. So I say solutions have less solute than the inside of the cell. And then in this case, if the salt concentration was if the amount of salt here was higher, and then the amount of than the one in the cell, water will start moving out of the cells to equilibrate. And as the water moves out, the cells would shrivel up, crenation. So I kind of switch this stuff up. So take a note of this. I talked about facilitated transport, material moving through this transmembrane protein. Active transport is almost like the opposite of diffusion. Because in diffusion, we are counting on molecules to move from where they are in higher concentration to go to where they are in lower in low concentration. Okay, let's examine the situation here. This is inside the cell. This is outside the cell. These ones are sodium ions, potassium ions, potassium ions. And then if you look here, there are sodium ions and sodium ions. If we're following the idea of diffusion, would these sodium ions be moving into the cell or outside of the cell if you were just considering diffusion? If we were, again, my question is, if you are understanding that diffusion is when molecules move from an area where they are in high concentration to an area where they are in low concentration. I said, forget about the labeling here. Just imagine that we're talking about diffusion. Would you expect these sodium ions to be moving inside, or you would expect them to be moving outside of the cell?
Inside?
They will be moving inside, because here they are in high concentration. But in active transport, the reverse happens. Because inside, there are some ions or some molecules that the cell doesn't like to have them inside the cell. They want them in lower quantities as much as possible inside the cell. Some of them are sodium ions. So even though there are high amounts of sodium ions here outside the cell, if the amount here inside the cell is still relatively lower, sometimes the cell will still need to push some of these sodium ions out. So they are pushing them against the concentration gradient. And to be able to enable the cell to do this, the cell requires energy to do that. And how does the cell have the energy to do this? It breaks up ATP, adenosine triphosphate, to release energy, which will enable the cell to push these sodium ions out. And then for large molecules, sometimes the cell will want to bring a big chunk of something or many molecules into the cell. They don't go through like one at a time. The cell can just it folds. It's almost like you're depressing a round ball that does not have enough air inside, and then you put your fist inside. It creates an invagination, like a U dimple on the surface. And then, so in an area where those molecules are present, the cell will form this invagination, this big dimple, and then the neck of it, they will start coming close to each other, and then they will seal off, and then it will get into the cell. When they get into the cell, some things will break down the plasma membrane and then release the content into the cytoplasm. If these molecules are large solutes, they're referred to as endocytosis, or sometimes you hear phagocytosis, but in the case here, if they are liquid molecules, they would be referred to as what? If they're liquid molecules, they'll be referred to as pinocytosis, and then they'll bring them into the cell. Sometimes even the cell would have receptors on it, like in this case, receptors on it, that it will allow for selective binding of particular molecules And when they bind on it, the cell can now do the same thing and bring them into the cell and then release those molecules. Now, we went ahead and talked about the various functions, the nucleus, the command center of the cell, the ribosomes, the sites where proteins are synthesized, the endomembrane system. Just let me get a better picture here. Talked about the nucleus that has the nucleolus. Nucleolus makes RNA molecules, ribosomal RNA molecules, and then the nucleus itself, it has DNA, which is normally bound to proteins called histones. And then because the DNA is contained in here, the nucleus itself is referred to almost like the command center of the cell. And then from on it here, you have this wall. The nuclear membrane is also made of 2 layers of phospholipids. The outer layer folds and forms these foldings, and in certain portions, as it is folding coming from the nucleus, we have these yellow dots on it that are painted here yellow for illustration. Those are the ribosomes, and it is where the proteins are synthesized. And this folding continues to an outer portion here, getting towards the cell membrane, which the same things are happening, but now the ribosomes are not here. And these are smooth endoplasmic reticulum, usually involved in lipid metabolism. Then we talked about, so you have the same architecture here, the nucleus, the nuclear membrane, its foldings, the smooth endoplasmic reticulum. And when material is made, whether it is lipids or whether they are different kinds of proteins, they usually go and to this structure here called the Golgi apparatus, where they are packaged. Basically, when they say they are packaged, certain things might be added to it, and then they are formed, they're enveloped in a vesicle with a membrane, and then sometimes they can even go now and do the opposite of endocytosis, which is exocytosis. And then when the membrane that is binding them fuses with the plasma membrane, and then it releases the molecules out. Then this is all what I've been talking about. Then the cytoskeleton, we said inside the cell, it is not just like empty or pure water. They have like fibers in there, different kinds of fibers, some of them thicker than others. So you have, they said, large microtubules, thin actin filaments, and then medium-sized intermediate filaments. All of these things tend to hold some of the organelles together and then also give the cell its shape and its structure. Some of these fibers actually can even be very long, and we see those to have like when you see like a tail of the sperm tail, the long tail is made of some of these fibers, and in this case, the sperm cell now use them for motility. And then we said, when you look at cells, besides red blood cells that are moving by themselves, the rest of the other cells sometimes they are connected to other cells. And what is the manner in which they are connected? I said there are 3 main types. So I said adhesion junctions mechanically attach adjacent cells, almost like those of the skin, to make sure that it's a little bit very sealed. Meanwhile, tight junctions have connections between the plasma membrane proteins of a neighboring cell. We talked about proteins on the cell membrane. So just imagine 2 cells sitting next to each other. There is some kind of a connection between the transmembrane proteins of one, linking them to the other transmembrane proteins of the other one, forming like a zip-like connection. Gap junctions, on the other hand, it's almost like they are tubes connecting one cell to the other one, and it helps for movement of material, exchange of material between both cells. Adhesion junctions, tight junctions, gap junctions. Then we went ahead and talked about the plasma membrane. The mitochondria main function is to generate ATP. It has two membranes. You have this outside membrane, then you have the inner membrane here that also folds. And in this inner membrane, you're going to see that there's a lot of chemical activity going on there with certain things that would help in the process of generating ATP. The cell is believed to have gotten this organelle from the outside. Why? The belief is why? Because again, it's almost like the size of a bacteria, and it has its own DNA that is separate from the one in the nucleus. So we went ahead and I think we stopped somewhere around here. Then I wanted us to talk about enzymes, because the lab we have today has to do with looking at how enzymes work. What are enzymes? They are basically proteins that act like catalyst. What is a catalyst? Something that speeds up the rate of a chemical reaction. So a chemical reaction is like you have A and B being the reactants, AB being the product. So in a chemical reaction, this reaction would happen if both things can actually combine. What a catalyst does is just make it convert A plus B to AB faster. So catalysts change the biological catalysts, and they help to speed up the rate of reactions. And then some of the reactions that we're going to see are like in cellular respiration, where the food we eat is converted to energy in the form of ATP. And we say, for example, in terms of their nomenclature, in their nature, first of all, in their nature, enzymes are proteins. So as proteins, the environment in which they are able to work has to be suitable for them, or else they can denature the proteins. If an enzyme is supposed to work at 37 degrees, like those in our body, if you put them up to around 70 degrees Celsius, they will not be able to work. Why? Because that temperature would make the enzyme to change its shape and it will not be able to work. I said some enzymes are aided by non-protein molecules. So we have sometimes like our vitamins or some of the vitamins we take, and there are some other molecules that just work together with the enzymes to make the enzymes do their job better. And how do enzymes work to make the reaction happen faster? They do so by lowering the amount of activation energy that is needed to make that reaction to happen. Take a look at this. So first of all, when you would hear the word metabolism and reactions, the way of referring to a sum total of all the reactions that happen in our body, they usually use the word metabolism. And then in this previous situation here, I showed you a situation where reactants would work A plus B to form AB. Sometimes it ends like that. AB is the product. But sometimes AB becomes the reactants on which different enzymes work on them to convert them to something else, which is what is being illustrated here. A is the reactant that goes to form B, and then B the product, and then B turns out the reactants to mediated by another enzymes to be converted to C, and this one next be converted by different enzymes to D, and it can go on. So you can sometimes you have a chain of reactions that happen, many steps to be able to end up with a final product that is desired for the cell. I said enzymes work by lowering the activation energy. If you take a log of wood, for example, and you put it next to you, it's not going to start generating heat, but there is energy inside that dry log of wood, which if you light it up with a lighter, it starts burning and then it starts generating heat. The lighter that you use it to light up the wood, the piece of log of wood, is the activation energy that is required to make that reaction of the fire burning the wood to continue and generate more heat. In chemical reactions, is something akin to that. If you imagine that every reaction would require some amount of energy to get it started, the lower the amount of the threshold of energy that is required to get it started, the quicker it's going to start. Some of them would require more energy. And so it would take a long time to keep putting in the energy before it starts, the reactants begin being converted to products. So you can actually graph, do a representation of how energy is involved in the chemical reaction. The y-axis here could be amount of free energy required, and then the x-axis is how the reaction is happening, meaning that how the reactants are being converted to product. There are two lines here illustrating two situations. If you look at the one in blue, you have here reactants. Let's say all the starting point, everything is here, and then you start the reaction to happen now, but you need to give it energy. For one reaction, the blue one does not have enzymes, and then the gray one or the gold one, there's an enzyme involved. So if you imagine here that you have to add some energy, the energy you add, it is increasing, and then the time it gets to the top here, the product starts forming as it starts coming down here. So if you look here, the delta from this point here to this point here where the products start forming, this is the amount of activation energy that is required for the blue reaction. You keep applying energy to this point where you don't really need any more energy. The product starts forming by themselves and going forward. Here, the same reaction, same reactants, one without enzymes, one with enzymes. The one with enzymes here, the difference is it starts faster thanks to the fact that it has enzymes to lower the activation energy that is required to make the thing to proceed. Now, how do enzymes work? Enzymes have a protein in nature. They have active sites. So in terms of the protein, the enzyme, they have like special grooves or certain areas where the substrate will go and fit inside. So the reactants will fit into that. It's almost like it's almost like you can look at it like matchmaking. Imagine that everybody's sitting in you have people in a big room and finding out who they can connect with. Imagine that is the situation that happens when there is no enzyme involved. But imagine that you have a matchmaker that is inside the room, knowing that red would like white or blue would like pink. That matchmaker can easily pull out who is blue and who is, and make sure that they grab their hand and take it over, and then grab the hand of the other person that they will match, and then they hold their hands and let them connect. It's something akin to that. This enzyme is the matchmaker. The substrate comes in and the right substrates come and are grabbed and held, and then connections happen. And then when the connections happen, then the enzymes says my job is done. Then the matchmakers now continue forming their union or disunion, and then the enzyme continues, goes back and does the same matchmaking again. Enzymes have active sites. When substrates fit into those active sites, some things happen in there that makes the connection or the reaction between those reactants to happen so that the product can be formed faster. And when we talk about an area too where enzymes are involved, I said in our cells where we have cellular respiration to generate ATP from the food we have eaten, but even to the point at which our cells start dealing with this food, we have to eat the food. We have to break it down to the form in which we can absorb it. So it is thanks to the enzymes that help. This is our stomach right here. When we eat food from our mouth, digestion has started for carbohydrates. Digestion starts with what? With enzymes. In our mouth, we have salivary glands that act on the sugars that's acting on carbohydrates in our mouth, come to inside our stomach. We have pancreatic juice that are secreted by gastric pits in there. Gastric juice that are produced in the presence of hydrochloric acid that lowers the pH. Some of the enzymes in their inactive form, like pepsinogen, would get converted to the active form pepsin, and it starts acting on proteins. Then you come here, a bunch of enzymes are happening here. The point here is what? Our process of digestion and absorption happens thanks to enzymes. And to talk about a little bit about cellular respiration, it is good to kind of look at how plants even make carbohydrates at all. Because cellular respiration is almost like the opposite of a process called photosynthesis. Photosynthesis happens in green plants. What does it involve? Carbon dioxide in the air combining with water with the help of enzymes to form a glucose molecule. Green plants do this. Why? Because, and they require what? They require sunlight in this process. So carbon molecules are combined with water molecules connected together by harnessing the energy from the sun and then forming this glucose molecule. The light energy which is used now to create this bonding to form this larger molecule. The byproduct of this process is oxygen. So here we have an apple that has been created just from carbon being gotten from the atmosphere, water pulled from the soil, forming apples which we can eat. When we eat the apples, the reverse happens. This is the apple we are eating. that is the glucose, we need to break it down to release energy. And the end products is carbon dioxide and water. Let's watch. So this is the process of respiration. We need oxygen in this process. We are eating carbohydrates like the apple. We do the digestion part of it with our enzymes. We absorb the carbohydrates in the form of glucose. And now we have to now convert that glucose into energy for us. So we can thus far know how we get the food material, the apple, from the store or from the tree. When we eat it, enzymatic activity happens for us to convert it into glucose, the form which we can absorb. After we absorb it now, let's see what happens. The glucose gets into our blood vessels, start moving around. The cell in the neighborhood says, I need some more energy. Then, because the glucose molecule is now, they will move from the blood, come into the space between the cell and the blood vessel, and is now taken into the cell. When it moves into the cell cytoplasm, a bunch of activities start happening again thanks to enzymes. There's a conversion factor here that takes this and breaks it down. As you're going to see, glucose has 6 carbons. See, it says 6 carbons, 12 hydrogens, 6 oxygens. One of the things that I want you to think about is, what is the function of oxygen? Why do we breathe oxygen? The reason we breathe oxygen is for this process to happen. Now, how does the process really happen? To the next. The goal of the reaction is to break 1 carbon at a time, mix it with with oxygen to produce carbon dioxide and water. In so doing, releasing energy. As the energy is released, the question you should ask: how is this energy retained? Or is it produce it and use it right away, or there's a way to store it? The answer is there's a way to store it. This entire process has been broken down into 3 steps for illustration. It is a continuous process, but to facilitate understanding, you can look at it in phases. The first phase we can refer to it as glycolysis. Glyco is the sugar, lysis is breaking it up. The citric acid cycle is the next step. Because it's a circle, means that a molecule comes in, goes around and around and around, and comes again to where it begins, and then some other molecule that is accepting the glucose molecule does the same thing over and over. Then you have the electron transport chain. It means that there are a bunch of electron transporters. One takes one electron, hands it to the next one, the one hands it to the next one hands it to the next one. The question is, why does this happen? What is the benefit? And where does it happen? So we're going to take each of these phases and look at where each one happens. Glycolysis occurs in the cytoplasm. It breaks down the glucose molecules into 3 carbon molecules called pyruvate. In this breakup system, hydrogens are released, like protons are released. Hydrogen ions, like H+, are released in this process. This H+ traps carries electrons. They use electrons to connect. Electrons are produced that are captured and held by these molecules. They call them NAD. They use electrons to involves this. It has like this NAD happens by itself. This breaking into pyruvate happens, and then it releases electrons that take a hydrogen and connects it to this to form these molecules. In also so doing, some of energy is captured and 2 ATP molecules are produced. We'll look at it. But the goal here is, where does it happen? What happens? What are the starting materials? What are the end products? The citric acid cycle, also called the Krebs cycle after the guy who probably discovered it. Where does this happen? It happens inside the mitochondria. I say it is a cyclical pathway that occurs in the mitochondria. I say it also produces NADH and it also produces ATP. This step requires oxygen. Meanwhile, glycolysis did not require oxygen. The question is, what does oxygen do here? To answer the question preemptively, I said oxygen serves as the final electron transporter. When all those electrons that are being carried keep moving by those transporters, the final acceptor of these electrons is oxygen. When it accepts it, it takes hydrogen. Two hydrogens are connected to it to form H2O, which is the water that comes as a result. And then you come here in the electron transport chain. I said this is a series of molecules embedded within the mitochondrial membrane. I said their job, as they move the electrons, they are being captured. Electrons, some of them are carried by this. I said NADH is made in step 1, and 2 carry electrons here. The total of 32 to 34 AT molecules are made in this final step, depending on which carriers are involved. Okay, so let's take, I'm going to move here. So I kind of use these pictures here to kind of illustrate this. Normally, they would illustrate this like glucose molecule in this fashion, a ring structure. To make it easier to understand, I put these black dots here to be the carbons. Remember that these carbons were formed by one carbon dioxide being combined with water to form, to connect the carbon to the next, so that they form the first glucose molecule. So as this step, the first step I said will involve breaking this 6-carbon structure into 2 3-carbon structures. I put here that you require some ATP to do it. However, at the end of that, you have a net amount of ATPs are produced. You require activation energy to start this process. But let's say you started with 1 ATP, you started with 1 ATP molecule as like activation energy. If you end up with 2 ATPs, it means that you have a net of a certain amount of ATPs. So the first step here is, of course, in the cytoplasm, breaks this into 2. The products that are produced is what? NADH, which carries electrons. This is an electron carrier. And then here, so you started here, 2 ATPs go in, 4 ATPs eventually are produced, but the net result, you see how it ends. So these pyruvates, they'll call it pyruvate, and you sometimes see some books refer to it as pyruvic acid, is the same thing. You don't want this to stay too long in your cell. Now, to move on to the next step, this step here, this would lose one carbon, and then create a product that would enable it to get into this into the mitochondria. I say the citric acid cycle, called the Krebs cycle, is cyclical, occurs within the mitochondria. Its products, electrons and ATP. So you have here this material here, this 4-carbon molecule, existing within the mitochondria. Now, lose one carbon to form carbon dioxide. The product that results from it is called acetyl-CoA, made up of just 2 carbons that are left. These 2 carbon products that came from pyruvate will combine with this 4 carbons to form a 6 carbon. So you imagine that this one comes into the mitochondria, and then it's welcomed by this one. They form this structure here that has 6 carbons. A reaction happens to convert it into this, losing 1 carbon. And then another reaction, this becomes the product here, and then it loses another carbon again to form the 4 carbon again, and then which is the original material that was present. In so doing, in each of these conversions, electrons are released that are carried by an electron carrier. Carbon dioxide is produced as well. And then I mentioned that the electron transport chain happens. Where does this thing happen? I showed you the inside of the mitochondria, that the inside part is kind of folded. It's folded to increase surface area. I said the electron transport chain, series of molecules embedded in the mitochondrial membrane. These are the mitochondrial membrane. This one here, it's a lipid bilayer. This is the membrane, and then you have these electron carriers, these blue things. Their job is to take the electrons that are produced from this conversion here. Look at here. I said each time this one cuts a carbon out, electrons are produced. Cuts another carbon out, electrons are produced. When they produce the electrons, what happens? The electrons are carried by these folks, and they will carry them and hand them to this one, and hand them to this one, and hand them to this one, until they come here, and then they said the final one that accepts it is oxygen, and it uses it to attach hydrogens to it to form water. As these electrons are being carried out, something is happening. You would have protons being moved into the intermembrane space. Protons, like hydrogen ions, as they are handing out, hydrogen ions are moving here, are moving here, are moving here. Now, you cannot see when the concentration of the hydrogen ions, which are also referred to as protons, build on this side. The only way they can get back to equilibrate have equal amounts on this other side is that they have to pass through a certain transmembrane protein. This particular one is called ATP synthase. ATP synthase. As the hydrogens they build up here, as they see that this is their only channel to move out, they start flushing out. As they start moving out, this is almost like a small generator. As they do this, ATP molecules are being formed. And when they come out here, they will combine with the oxygen, with those electrons that the oxygen has to connect them to form water. And as they're so doing, they produce ATP. In this entire process, all I want you to know is electron carriers exist here. Protons are pushed this way. The protons would eventually move through this ATP synthase. As they go out here, the oxygen we breathe now we'll take those electrons and then combine it with hydrogen ions to form water. The total in this process, about 32 to 34 ATP that are being formed. You could watch this, because of the bandwidth I have, I don't want us to play an animation of this, but you'll be able to see this, and you can play an animation of this. You will see it illustrated very well. Now, the question that you can ask is, does it mean that you can only produce ATP in the presence of oxygen? The answer is no. Fermentation is a process where you can get ATP from oxygen from ATP from carbohydrates without oxygen. So the process of fermentation that happens, or where you can get energy, ATP from oxygen, from carbohydrates within our cells, is that step of glycolysis, because it doesn't require oxygen. But only 2 ATPs are produced in that process. Right here, we go back here. Right here in this step from here to here, a net 2 ATPs are produced. Even though, imagine the fact that if 2 ATPs are produced here, and then this other process that happens inside the mitochondria produces 32 to 34, it means that this product of pyruvate still has a lot of energy. But sometimes, when does our body actually a sprint, or you have to do something that requires a surge of energy, your cells have this capacity of exhausting, trying to look at all the carbohydrates that are readily available and trying to convert them into pyruvate to generate this ATP so quickly. They are hoping that as they do this, the pyruvate generated can be easily converted into the form that can get it into the mitochondria. But if something happens and you are in a situation where there's not enough oxygen coming to those cells, this pyruvate starts building up. And when it builds up as it will get converted to lactic acid, to lactate, which is sometimes they'll call it lactic acid. When this is converted to lactic acid, the way we experience it is like a muscle cramp, sharp pain, because that acidic pH the muscle cells or cells don't like it, except those in our stomach. So let me ask you, when somebody has a muscle cramp, anybody?
I think usually people will stretch to get rid of the cramp.
They stretch to get rid of cramp, or sometimes they'll come and massage it to try to get rid of it. What does that massaging process or stretching, what does it do, you think?
Maybe it like helps. Yeah.
It does what? It relaxes the muscle and brings more blood to it. As the more blood comes to it, you will see now that, oh, wow, that's great. Now, blood is bringing oxygen, and as it brings oxygen, your cells are able to convert the lactic acid back to pyruvate to make it go through the mitochondria process for cellular respiration. So that is the benefit of that. Now, so again, the thing that I want you to know here is these 3 steps of glycolysis, the Krebs cycle, or and then the electron transport system. Where does it happen? What happens? What are the products that are involved in it? What are the reactants that are involved in it? And what are the products? When you look at it from that vantage point, that is really what I expect you to know in this process. And to be able to help you do that, if you watch the video of how this is happening, then that would be, and then you look at these notes with these summary notes, things can become much more, they'll make more sense. And if they don't, let me know. So far, does anyone have any questions? Any question? I take that as a no and push ahead. So this is kind of like a summary slide that talks the whole story. Glucose comes out of the cell, of the blood vessel, carried by a transmembrane protein into the cytoplasm. Into the cytoplasm, it undergoes, this is the first step, glycolysis. Product, a reactant is glucose. Product is pyruvate. For the pyruvate to get into here, this pyruvate 3-carbon, it will lose 1 carbon, like carbon dioxide, and then the 2-carbon molecule comes into this cycle, the Krebs cycle or the citric acid cycle, and it attaches to, like part of the molecules in this cycle, the one that it attaches to first is the 4-carbon molecule, then has a 4 plus 2 is 6-carbon molecule formed, and then it gets converted into a 5-carbon, then the 5-carbon then goes into the 4-carbon again, it comes again, it takes the second pyruvate molecule, the second acetyl-CoA molecule, and does the same thing. Here, look at the products that are being produced, ATPs, the number of ATPs, the number of ATPs here. So if there's no question, I would like us to push ahead and look at the next, give me 1 second here. I would like us to push on and look at the next chapter, which for our lab today, we're going to do appreciating how enzymes work, the properties of enzymes, and how they we've talked about what a cell is. I think it would be now proper to look at how cells operate. Do they operate by themselves, or do they operate, or how do they even operate, by themselves or together with other cells? But I think some of them operate by themselves or would operate along with other cells. So I would say we go now to what we have as a organization and regulation of body systems into tissues. So most cells would work with other cells to perform the same job. Cells that work together, similar cells working together to perform the same job, is called a tissue. So we'll look at, I put here, I think let me do this. Let me bump this up into, okay. So I said, by the time that we are done, you now see how we've talked about organization from atoms to molecules, molecules to forming macromolecules, and then macromolecules and all that forming cells. So a cell, we defined it again as the basic unit of smallest basic unit of any living organism that can carry the processes that we described as said living things do. Now, we're going to look now how these cells are combined to form tissues. And we're going to look at the 4 major classes of tissues to look at what each one is, what kind of cells they are made out of, and what they do. Then we'll look at the relationship between cells, tissues, and organ systems, distinguish some tissues like the muscular tissue, the nervous tissue, and then we'll look at that and look at the skin, and then look at skin cancer and homeostasis. So what is a tissue? A collection of cells of the same type that perform the same function or common function. There are 4 main types: connective tissue, connect, it binds and support body parts. Nervous tissue, it moves the body and its parts, helps in communication to enable movement of the body and its parts. Epithelial tissue, it lines surfaces and cavities within the body. And then muscular tissue, it also helps to move the body in its parts. So nervous tissue helps for communication, and then nervous, this one, muscular tissue, also movement. That if you look at any tissue, the question you should first ask is, okay, what are the cells that are making up this tissue? And what does it do? If we take the first one, connective tissue, I say it binds and supports body parts. The tissues are usually, the connective tissues, they usually are found within a matrix. Sometimes the matrix can be liquidy. Like in this connective tissue, the matrix is semi-fluid, or sometimes it is solid. And then some of them might have protein fibers in like protein strands of different sizes. There are three main types of connective tissue: fibrous connective tissues that are loose, adipose tissue like fat tissue. Fat adipose tissue is a loose connective tissue. And then you have like dense connective tissue that you'll find them like in tendons and in ligaments. So our adipose tissue, like the fats that we have in our body and like body's surfaces, or like in our in surrounding the different kinds of some organs and certain parts of our body, and then in our lungs that have very small strands, have connective fibers of different sizes. They make up fibrous connective tissue. Then you have supportive cartilage. Cartilage is much more dense, is stronger, but flexible compared to bone that is more compact and stronger and less flexible, or sometimes not even flexible. And then the fluid connective tissue, which is made of blood and lymph. We'll take each one and look at it. So if you look like within our knee area, for example, and you could see here that in certain areas of our skin, you have a representation of these tissue types. So what they did here is, if you take, this is your knee region here. If you take this area around here from where the joint is here and the kneecap here, and then going this way here, if you take this and blow it up, you're going to see that it has various amount of fibers in there, like loose and fibrous connective tissue. You have elastic fibers, which by their name means they can stretch. We have collagen fibers, which are thicker but are not stretchable. And then they also have cells in there, which are like fibroblast cells. Now, if you turn around and look and say, oh, there's this bone right here, and this bone of your leg right here, those bones, they're kind of, something is holding them connected together. Yes, there is something holding. Ligaments connect bones to bones. Meanwhile, if we have these muscles here, our thigh muscles are connected to the bones to anchor them there. The connection at the end here, they are connected by tendons. And then if you look just here again, we talk about loose connective tissue. Right here again, there's adipose tissue, fat tissue here. And then by the bone itself, is compact. Here you have like fibrous bone, but the bone is stronger, more dense, not flexible when compared to what? Look at the end of the bones here. You have this white material here, which is cartilage. It's softer. It helps more cushion and more flexible for our bones so that they don't crash and grind on each other. It's like small shock absorbers at the end. So we can move here and look at, again, loose connective tissue. Where do we find these? Like in our lungs, in our arteries, in the urinary bladder. And again, some of them will have this kind of material and then they would have like, again, adipose tissue, same thing, ligaments here, tendons. Meanwhile, like cartilage. So where do you find cartilage? Cartilage, I said, is supportive connective tissue. What does it do? It supports stuff. But is all cartilage the same? No. If you look, I said there are basically like 3 kinds of cartilage types. So you have hyaline cartilage. If you touch your nose, it kind of wiggles a little bit. And then the ends of our long bones, the one I was showing you there. And then as the skeleton, as the fetus is developing, even the whole structure of the skeleton is made of hyaline cartilage to begin with. And some parts, like our backbone, will eventually get impregnated with calcium salts or things like that to harden out to become compact bone. But the point about here is hyaline cartilage is made of collagen fibers. And we also find it in of long bones and in skeleton and in kind of wiggle it, it's going to move. It's going to wiggle back and forth. It's because the material that is made of elastic fiber. It has a lot of the cartilage there is more flexible. In all this material, the cells are found within these small chambers called lacunae. Then the other cartilage type is like the fibrocartilage, much, much stronger relatively. Where do we find this? We find this in our vertebral column. Like our backbone here, you have the vertebral disc, each bone connecting to the other bone, connecting to the other one, connecting to the other one. So separating them is this cartilage. It cushions, you can make a headstand, you can jump up, you can carry something heavy, it allows you to bend front, back, lean backwards and things like that. It gives for flexibility and causes the bones of your backbone not they act like a shock absorber. They don't cause them to grind on each other. Bone, again, the bone cells are found within this matrix, this chambers called lacunae. If you look here, it's almost like if it was just one of them, it would look like how you cut some trees and then you see those circles in the tree. But in this case here, if you take this bone, you cut it into 2, and then take this surface, you blow it up, these round rings here. In the middle here, you have the lacunae, you have the blood vessels, and these concentric circles here have certain areas they're called lacunae in which you find cells. So even though you might look at, you cut a bone, you break it up, you look at it, oh, it looks like it's just like some piece of nothing that has cells. It has cells. It has a few blood vessels in there. As the cells are in chambers called lacunae, the matrix is solid and rigid, made of collagen and calcium salts. It starts with cartilage, but these areas get impregnated with calcium salt to make them rigid and hard. Compact bone made up of repeated circular units called osteons. And then within it has the blood vessels. And then there's for this, like this long bone here, it has an empty shaft inside that might have bone marrow. At the ends of this, you look at here, there is more of a spongy bone. It have like a scaffolder with spaces in there, a lattice. Now, again, so we talk about 2 kinds of supportive tissue, bone and cartilage, comparing both bone stronger than cartilage, less flexible, 2 types, spongy and compact bone. For cartilage, 3 types, hyaline cartilage, elastic cartilage, and fibrocartilage. Now we talk about fluid connective tissue, blood, and lymph. Blood is made up of a fluid matrix. So if you consider the liquid portion of blood, we call it what? Plasma. And then it has cellular components, which are what? They're sometimes referred to as formed elements, made up of what? Red blood cells, white blood cells, platelets. With the red blood cells' job, what? Carrying oxygen. White blood cells' job is to fight disease or fight infection or to get rid of bad cells in the body. They will have platelets, which are pieces of cells or cell fragments that help in the clotting process, blood clotting process. You have you scratch your leg or your hand, it starts bleeding. Thanks to these little fragments called platelets, they go and aggregate in the area where they created there, so that the blood doesn't come out anymore. You know, when you have a scratch on your body or a wound, you see it as red because blood is oozing out. Red blood cells are coming through. When the platelets go and cover them, and the size of the cover, the aggregation, out, then you are not bleeding anymore. When you have a scratch and then you go and say, oh, put your finger, press and hold it tight for a certain number of minutes. When you press and hold it tight, what you are doing is you get these platelets to come and build up there to form the mesh that will prevent the red blood cells from coming out. Sometimes when you have actually stopped bleeding, you can still see clear liquid coming through. Yes, because that is just the liquid portion, the plasma that is coming out. So what is the difference between lymph and blood? So I said lymph, we usually find it surrounding our cells, like it's part of the extracellular, some of the extracellular liquid that we have goes and flows and connects into this parallel network that we are going to say called the lymphatic system. This system does not have red blood cells. Movement of the liquid in this system, the lymph, is not controlled by our heart beating as it is with that of blood. What does it have? It does not contain red blood cells, but lymph does contain white blood cells. So in summary, what do I expect you to you have? Summary of the types of connective tissue, we have it here. And then you have here all of this, adipose tissue, white blood cells, and then we have collagen in fibrous connective tissue, and all of this here. You have almost like a schematic here that tells you the three different kinds of connective tissue: the fibrous, that's made of the loose and the dense, and then you have supportive, that is cartilage and bone, then the fluid made of blood and the lymph. Then you have muscle connective tissue. What is the job of these muscles? It allows us to be able to move. And what is it made out of? It's made of muscle cells or muscle fibers, along with protein fibers called, which are like, some of them are actin, some of them are myosin. There are three types of muscles that we have. We have skeletal muscles, those which are connected to our skeleton, and then we have smooth muscles, some of them found in the walls of our blood vessels, then cardiac muscles found in the heart. We'll take each one and look at it. Skeletal muscles. So I said, in appearance of skeletal that had only just one nucleus. These ones have multiple nuclei. And they have, if you look at them, it looks like they have some dark and lighter bands. If you look here, there is let's do something here. So try to get at what is it? Let's keep going with this cursor type. So you have darker bands here that are brown or reddish brown, and then in between them, less brown side. So these areas of darker and lighter bands are called striations. And striations, if you look at them, basically what they are, if you put your fingers in front to interlock, the tips to interlock, if you would notice that the areas where they just kind of interlock, they are, because of the interlocking in that area, it's almost like a darker strand. And then at the end of the beginning of your fingers tips, the gaps on your hand, the gaps where your fingers are starting from, they have holes that are visible. So these areas here are the areas where the fibers are interlocking, because the fibers tend to contract. When your muscle contracts, these fibers shorten. And then when they relax, they extend back. And then these fibers would form bundles. Again, in terms of how they are located, they are attached to the bone, and they help us to be able to move. When they contract, we can move, we can stretch our arms, or they help us to pick up something or even move from one place to another. Then smooth muscles, I said. And a cross section of our blood vessel. This is an epithelial lining in the middle called the endothelium. And then here underneath it, we have these smooth muscle cells. And since they are smooth muscles here, and they call them vascular smooth muscle cells in this area. In terms of appearance, how do they look like? They look like almost like a croissant or something like that, where you have the ends are tapered, but the middle is bigger. They don't have striations. They have just one nucleus. The way they contract, and they do contract, but their mechanism of contraction is different from that of our skeletal muscle that involves shortening of fibers. And contraction of the smooth muscle cells is actually, we are not conscious about it. Imagine that you want to pick something up. You could decide and say, I want to pick this pencil up. Say you stretch your arm. Your brain tells your muscles, the biceps, to either, when your hand is stretched, it says, okay, contract, I want to pick this up and give to me. Your biceps contract, and the other side again, the other opposing muscle set relaxes, you're able to pick up something. So you are conscious of doing that. You want to do something, and you tell yourself. But as you and I are sitting and talking, blood is moving in our blood cells and in our blood vessels. We don't know when it's contracting. So it's almost like we are not aware of it. So we refer to that kind of action where we are consciously not aware of it, involuntary movement. So in hollow organs and blood vessels, we find these smooth muscle cells. Now, cardiac muscles. Cardiac refers to the heart. Make up the heart. The funny thing about it is they have characteristics that are similar to the smooth muscles and similar to the skeletal muscle. So let's look at, they have, the cells, as they have a single nucleus, just as the smooth muscle do. They have striations, just as the skeletal muscles do. But they also have like segments, like here they have, they call it intercalated disc, besides these striations here. And they also, I just said, they have a single nucleus. Where are they located at? Definitely, since it's cardiac, they're located in the heart. Also similar to the smooth muscle cells is that we don't tell our heart to beat. It just starts doing its thing. And actually its control, or what even makes it to start beating, is intrinsic. What makes your heart to start, we will see some cells, some areas that actually produce electrical stimuli that will make the muscles to contract or relax. We'll look at that. So in talking about muscles, it probably would be good to also mention some disorders. Like, for example, if you use your muscles a lot, you might have strains, or you might have sprains or cramps. I mentioned muscle cramp before, where you are feeling pain in your muscle. Tendinitis, when the tendons are inflamed. Then you have disease, genetic disorders like muscular dystrophy, something that happens that messes up the connection or the innervation of the muscles, making them not to be able to listen to instructions to do what they're supposed to do. Cancers, when you have some of these cells start growing in an uncontrollable manner. And then inflammation, called myositis. Myositis is when the muscles are inflamed, and then we have, again, disease of nerves that affect muscles. Again, you could have receptors on the muscle neurotransmitter that will go and make them to send signal for them to be able to perform. And then you have infections that can cause problems. And then we have some medicines that could also cause problems for your muscles. Now, in terms of nervous tissue, it is made of what? Nerve cells. We cannot break the things up into, we have, it has a whole tissue that is made of nerve cells. We'll take what kind of nerve cells are involved. This job is to allow for communication, to pass information. We see from our sensory organs, internal and external. It connects, it captures this information, takes it into our brain for understanding, integration, they call it, and then relay it back to our muscles to do something, or some parts of our bodies to do something about the information. So we have two cell types involved here. We have neurons, excuse me. We have neurons and neuroglia cells. And you can even see that the shape of the neuron, for example, you can imagine that its shape makes it suitable for what it does. If it was just round, it means that you need many round things to be packed up, like some of you were trying to count the number of red blood cells in the field of an electron light microscope. But look at these ones. They are long. They have a head region here that has a nucleus, and then they have this long, almost like a long tail here. And then it has these portions here called the dendrites. It's almost like antennae that will pick up signal. When they pick up the electrical signal, when they pick up here, the signal here, they have something they call the action potential that is generated. It moves, that signal moves in that method through this long tail here called the axon, and ends in this area that have like buttons on it. A broad finger-like extensions or stretch-like spikes around here, dendrites, to be able to pick up sensation from a wide area. Like any cell, it has its own nucleus too. But also here, it's almost like you took like it's almost like you took a sausage and connected into many sausages. But see, the point here is there is some covering material here, and then another covering one here in like there's have gaps in between. These material and then these gaps here are called the nodes of Ranvier. Maybe after they do who discovered it. I said neurons are made of dendrites and a cell body and an axon. Axons carry information away from the cell, from the cell body. Then they have, so the main cells here are neurons. Neurons take information from our eyes, from our ears into our brain, and then our brain makes sense out of it, sends out information out with instructions to go do something. For example, you look at something, information comes from your eyes to your brain, then something is flying and coming towards your eye, and then it goes from your brain to your eyelids and say, close your eyelids, you don't want some of that insect to come into your eyes. So a certain set of neurons that are taking information to your brain or your central nervous system are called sensory neurons. The neurons that are carrying out information from our brain or from our spinal cord and going back, say, to our muscles to tell them to do something, those neurons are called motor neurons. Meanwhile, all these neurons have supporting cells. Some cells whose job is just to make sure that they are doing their job properly. So some of the supporting cells, supporting neurons, are called neuroglia cells. Some of them are like astrocytes, oligodendrocytes, and microglia cells. Then we go to the other one, the last one. I said the epithelial tissue. I said a cell group of cells that form a tight, continuous network, either covering the insides or the outsides of our body. Lines body cavities, covers body surfaces, and found within glands. The thing about them is that their cells are anchored, they are attached to a basement membrane on one side and free on the other side. If you think about it, like our skin cells, epithelial cells that cover our skin, one side of the cells is free to the outside, right? And then the other side is attached to the below, to the, I have a base area which is attached to. Cells are anchored by a basement membrane on one side and free on the other side. They are usually named in the way they appear or the number of. Sometimes you would have some epithelial cells that would change their shape just because of certain stresses or tensions being applied to them. So I said in terms of naming, you could name them based on the number of layers, if it's just one layer. And I said, typical to them again, they are attached on one membrane, a basement membrane, and then on the other side, they are free. So based on attachment, you could see here, attached on one side to a basement membrane, attached to a basement membrane, attached to a basement membrane here. And then one side is free. This side is free. This side is free. This side is free. They could be one layer. When they're only one layer, they're simple. When there are multiple layers, they are stratified, means there are multiple layers, like the ones of our skin. And then if they kind of look like they are stratified when they are not, like these ones, if you look at this, it looks like as if they are multiple layers, but technically it's just one layer. You have this one attached to this one, but tapers at the end. This one attaches to the here, but doesn't really show up. And then, so it looks like here it is stratified, but it is not. When it just looks as if it is stratified, they refer to it as pseudo stratified. And I said you can also name them too, again, based on their shape. Based on shape, some of them will look like cubes, some of them will look like columns, some of them will look like flat. Simple one layer. Look stratified, pseudo. Stratified, many layers. When they are flattened, they call them like squamous epithelia. When they look like cubes, they refer to as cuboidal epithelial cells. When they look like columns, they refer to them as columnar epithelial cells. We see some examples here. Lining of our small intestine. Simple layer, pseudostratified, ciliated columnar epithelia. Look at the words. Pseudo-stratified, it means it looks like multiple layers. Ciliated columnar, they are columns. That means they look like, they're not cubes. A cube has all the sides the same, but this one, they're like columns. And then ciliated, it means that on the edges here, they have cilia, which are like hair fiber. They look like this is the basement membrane. This kind of makes sense for what they do, because this is our trachea that leads from our nose into our lungs. And the purpose of these cilia is that if you are inhaling material that has particulate matter in it, this cilia will trap it and then whip it out so that it can get out of it, and then you can cough it out. Stratified epithelia, lining our mouth, for example. If you see how you were able to get a sample from your mouth, when you scraped a little bit without poking it, you're able to peel some cells from it, and then you were able to stand them and examine them. Our mouths keep sloughing. To really appreciate this, if you make a mistake and you drink coffee that is too burnt, too hot, you can even see the part of the cells peel. But most of the areas that are stratified, they have constant exfoliation. So we have the ones of the mouth, the ones of the esophagus, and the anal canal and the vagina. All of those have stratified epithelial cells. A better picture here. Now, I just put a summary here for various cells that are involved and where you can find them and what they do. Now, I had mentioned earlier, we talked about how cells are connected together. Again, this is a reiteration of the same thing. Gap junctions, tight junction, adhesion junctions. And if we look at our skin, for example, the skin, I think, is something we should take a quick look at it much more deeper. So I said, if you look at our skin, it's usually referred to as the integumentary system. It has accessory organs like hair, nails, and various glands. The skin has two main regions. A portion here called the epidermis, and below which is the dermis. And then below that, you have a subcutaneous area that has adipose tissue, and you have blood vessels connected to it. I said, what is one of the main functions of the skin? It helps for homeostasis. It helps for us to be able to sense our environment. We'll look at some of those functions. I say it protects the body from physical trauma, invasions of pathogens, or water loss. Because it also helps to regulate our body temperature. When we sweat, we lose heat. It allows us to be aware of our surrounding through sensory perception. And even on the senses, that you have hairs on your skin that help you to do that. It also helps for us to synthesize chemicals such as melanin and vitamin D. Melanin accounting for our skin shade colors, the various shades of our skin pigmentation. This is a good structure, a cube of our skin. If you look at this, these are the hairs on it, and the hairs normally they would even when you are cold, when it's very cold and you have hair on your skin, thanks to what? You would see a relaxation of your contraction of your erectophilar muscles to make these hairs to stand up. And then when they stand up and they overlap at their tips, they kind of create like hold air below to form this air cushion, which is a poor conductor of heat so that your body doesn't lose heat much. Contrary, when it's too hot, your hair doesn't stand up. It kind of lies flat. You might not realize it because, again, we don't have too much hair on some people don't have enough hair on their skin, but if you do, you would see that when it's too hot, you start sweating, the hair kind of lies flat to allow you to be able to lose heat easily. Because when you do that, you sweat comes out through sweat pores. It's a sweat pore right here. Is the muscle that I was telling you that is the erectophilar muscles that would contract when it is cold to make this hair to stand up. When it is hot, it relaxes, the hair kind of just lies flat. So if you see here, there is the hair root right here. And then we have some glands here called sebaceous glands. Where are they? They produce sebum. Oil glands here, they didn't say these ones here, they call sebaceous glands. They produce sebum, which is they produce oil to nourish. That comes here and kind of make sure that our skin doesn't get too dry. We have area here of these cells that are epithelial cells that are stratified. They keep flaking, exfoliating continuously. When you go take a shower, and the kind of thing that I would even is if you look at, we say it makes us also be able to sense our surrounding. For example, if you take a certain spot on your poking it with something, the same spot, and then when you pinch it, the same spot, you have a different sensation. You apply heat to the same spot, it feels different. Why am I bringing this up? Because you can imagine how complex something like that could be, where you are taking the same area, applying the same sensation, and differentiating the stimuli that are and the results that are being produced. So it allows us to sense our surrounding, helps in homeostasis in maintaining our body temperature by sweating. And then I think I put this picture here. And then I said, the thin outermost layer of the skin is made of the epithelial tissue. The cells in the uppermost cells are dead cells and become filled with material called keratin, which makes them sort of kind of waterproof barrier. It also has certain cells, white blood cells that are called Langerhans cells, that help to fight pathogens that want to get into our skin. And then I said that it also produces melanin, producing cells called melanocytes. They produce melanin, which helps to do what? Protect us UV light from penetrating and damaging our DNA. Some of the cells would convert cholesterol to vitamin D. So sometimes talking about the skin, it begs the question to also mention skin cancer. Skin cancer is a situation where you can have either the epidermal cells or the dermis cells to start proliferating, means dividing in an uncontrolled manner. Many a times when the cells divide, and let's say, for example, you had a wound on your skin, when they start dividing, they touch each other and then they stop. So we have certain things that control when and how cells would divide and then number of. And when they pile on in each other and form like a bunch of them, they form like a tumor. So you would hear things like basal cell carcinoma, which is the most common yet least deadly form of skin cancer. Then the melanoma, which is the most deadly form of skin cancer, but is the least common. Just general, I think there are many things that they would, doctor's office, even many areas, common information saying how to prevent skin cancer, depending on the kind of pigmentation that you have. Some people, and the susceptibility to penetration of UV light. The lighter you are in skin color, the more UV light can penetrate through because you have less melanin. And the impact of that is when UV light gets through your skin, it could get into your cells and mess up with DNA. And sometimes when it messes up with DNA and the cell wants to divide, messed up DNA doesn't divide well. And if the messed up section is on a crucial, important area in the DNA, like a gene that is controlling cell division, errors can happen during division, and then out of a sudden, a normal cell has been messed up now to start dividing uncontrollably. So sometimes, if you, depending on your color of your skin, you stay out less, or you have protective skin colors, color clothes schemes to avoid penetration of the rays. And then you could wear glasses, you could wear suntan lotion, sunscreen, not sunscreen or suntan lotion, sunscreen. And then they have different qualities of sunscreens. Make sure that you have the one that stays, and you understand the duration, how long it is good for. And if you really get too close to what does the UV light really do to your DNA, part of the nucleotides in DNA is thiamine. Thiamines are very close to each other. UV light will help to form connections between them when they were not supposed to have them. And when those connections are formed, the cell cannot divide properly. And then the other thing to also avoid, to make sure that to watch out to not to get indulged in, to avoid skin cancer, is all the tanning beds. When you go to a tanning salon, if they're using, some people, you go in a box or in a cupboard or in something, and they beam you with UV light. Not good for you. They tell you, even if there's a disclaimer on it, to the point that even in the state of California and some other states, if you are underage, you cannot use them. You could also achieve the darker skin. If you go to some salons that they use like a spray or some kind of application that they put on your body, you can just come out looking glow the color you want. The other bad thing is that if those that do a good job are slightly expensive in such a way that if you go to the cheaper ones, you might hate yourself coming out of there. You feel like you never went there. Now, I talked about the dermis. Now, the dermis is this lower portion here that has all of this here, made up of dense fibrous connective tissue, contains elastic fibers and collagen fibers, contains blood vessels, and also many sensory receptors and glands. Now, we talked about the keratin being filled on some of these outermost cells to make them more they form like a surface where water cannot easily penetrate them. And then we have some of it forms hair and it forms nails. Some of the other accessory organs of the skin and why they're important, nails are derived from the epidermis that offers a protective covering. Hair follicles derived from the dermis that allows our hair to grow. Oil glands are associated with what? Hair, and they produce again the oils called sebum lubrication to lubricate our hair and skin as well, and helps to mess up some bacteria from. Just give me 1 second, please. Give me 1 second. I'm going to put you on a brief hold here. Excuse me, sir. Sir, yeah, I'm waiting for somebody. All right, sorry about that. Let me just do something here. Are you still there?
Yes.
Good.
Yeah.
Let's finish this before we. Okay, I'm going to admit Chance before we leave. Give me 1 second here. So let's see here. So I would usually say in terms of when you have when everybody, you know how your skin is. There you have like birthmarks, coloration on your skin, and some you just oops, you saw something that is slightly different coloration, and then you are not sure what it is. Many a times when you see areas in our skin that is different, then you start going, oh, oops, what is this? I would say, don't even try to diagnose it yourself and say, I don't mind. It doesn't. But it is that this time they can actually ask you to take a picture of it with their cameras and send it to them. They can just look at it under the camera and tell you whether it is something you should worry about or not worry about. Because like in every situation, the sooner they see this, the better. If this one appears in our dermis, by the time it shows, so which means imagine here, this is our dermis right here. If the cancer is growing in here, by the time you notice it up here, it means that it has grown a lot in here. So that's why they call these ones a little bit much more deadly, because when they start here, you don't see them. And if you see it at the surface here, and you say, no, it's kind of a small dark thing, it's no big deal. So, but if you just notice that it's different, you tell your doctor, they will resolve it for you quickly, because most of these now are very easily treatable. So you could see this is the ugly side of the whole thing. And they call, all of this, when you see these colorations, just as I say again, UV light would mess up your DNA in your cells. There are other things that could also mess up your DNA, could make the cells become cancerous. So the more you're aware of some of these things, the more you can stay away from it. The easiest one that everybody's aware of is UV light. And then the worst thing is deliberately going to expose yourself with the objective of trying to get a tan to these UV lights that are, that would cause problems for you. So I said tissues to organs and organ systems. I said an organ is two or more tissues types working together towards a particular function. Our body is made up of several organ systems that are made of different organs that work out together to carry different tasks for us. So you have like a cardiovascular system, our lymphatic system, our digestive system, respiratory systems. We're going to go through some of these organ systems here, most of them, and then try to understand them some more. So I think this is about. I think there's one thing that we should mention here. I talk about membranes that line our body cavities. You have like a digestive tract. It has mucus. Some of the cells they have produced mucous membranes, and a respiratory tract, a urinary tract, a reproductive tract. And then we have like serous membranes that line our lungs, our hearts, abdominal cavity. So you have like the area where our lungs, the space in which our lungs are inside, they call the pleural membrane. And then our stomach, like our intestines, that gap, that space inside, again, there's a membrane that it kind of flows, makes it smooth, that they kind of move inside, and they call the peritoneum. And then our heart too is also contained with this space, in this space that is lined with this membrane called the pericardium. And then you have our joints that have membranes that lines the cavities, that the free moving joints, like if you're a ball and socket joint, we have the core that was synovial membranes. And then you have membranes that cover the brain and the spinal cord, called the meninges. The ability to maintain a relatively constant internal environment, which the body always strives to make sure that the body's processes are happening well. You have the skin helping, and then you have the nervous and the endocrine systems, whose key job is participating in maintaining homeostasis. Nervous system sensing our environment, endocrine system using chemicals, hormones to be able to drive certain things, the body to do certain things. Changes from the normal tolerance limits result in illness and can even lead to death, which is the deal of your body has boundaries in which it would operate nicely without any issues. But if you go within those tolerance limits, you can make you sick, or you can even kill somebody. Again, I said all of these systems that I had mentioned before, nervous, cardiovascular, digestive, urinary, respiratory, endocrine, muscular, lymphatic, all of these systems, when they are all working together, you say, oh, I'm happy, I'm healthy, everything is cool. You are not in a disease or in a ill state. So our life depends on all of these systems working together. So you can actually look at it. We started from what? Small molecules combining to form larger molecules, combining to form cells. Talked about cells organizing to work together in groups to form tissues. We have these tissues that are present, different types of them present in certain combination in groups to form a particular job. And in those tissues together form like organs. Now you have these organs that there are different types, but some of them would act together like in a group to perform a function, like a digestive, to organ system. Like you have here now, you have a digestive system that is made up of a bunch of organs, many organs involved. So you have, and then there are many, how many of these systems, but if everything is working together, we are happy, we are healthy, and then we have a way in which we communicate for things to happen. When we're talking about homeostasis, we want things to be in a particular situations. When things are sensed that things are going in this direction in a particular situation, information and said, do these actions to go in this other direction to kind of balance things out. So you have like feedback, negative feedback, which allows for to counteract and say, stop doing something. For example, if your blood glucose level is too high, then information goes and said, listen, go and do something to reduce this. It has negative feedback because something sends it and says, go do something. So glucose, insulin, more insulin. The insulin goes and said, okay, well, I'm going to reduce this insulin, convert the blood glucose to glycogen. Regulation of body temperature. If your body senses that the temperature is going too high, it does things to bring it down. When the enzymes are doing something and their job is done, the enzyme production to drive that process, information is sent, don't produce any more enzyme, we are all done. But there are certain reactions or certain things that are happening in our body that requires that the more something is happening, instead of stopping it from happening, you promote it. It's like feedback, cheer you up. Hey, right on, keep doing this, keep doing it. A typical example, for example, is during childbirth, there is a hormone called oxytocin that is secreted to enable stimulation of contraction of the uterine wall, like the womb, the area where the baby was staying. Now they start contracting to push the baby out. When contractions start, or for the reverse it, goes to that organ producing oxytocin, say produce some more, produce some more. It produces it, contractions happen more of the contractions happen so that the baby can come out easily, because it's a stressful process, not only for the mother, but also for the baby. Also, ovulation during ovulation as well. They said estrogen a secretion that makes eggs to get released. When the eggs are being released, you would see that you want the thing to spike, the level of estrogen to increase so that the egg can be released into the fallopian tube, hoping for eventual meeting of the sperm to have fertilization happen. So far, I've talked to you about negative and positive feedback. You can look at it and look at examples of some more. We went through all some of these different kinds of tissues, and I think I will end here. Any question?
Yes. Yes.
Yes.
Excellent. Okay.
Yes.
So what I'm going to do, we're going to talk about the cardiovascular system now, and we see how far we can go with this before my people are still working. So we've talked about these organ systems, organs, about tissues. Let's look at our first organ system that I think we should look at, which is the cardiovascular system. So the cardiovascular system the cardio has to do with the heart, blood vessels to deal with it. I'm going to put it here on a slideshow for us to talk about it. So I said, what do I expect you to know out of all of this? To be able to identify the components that make up the cardiovascular system, summarize its functions, and explain the purpose of the cardiovascular system, as well as and the lymphatic systems. So what are the functions of the cardiovascular system? What is the anatomy? What part is it? What is it made out of? What are the components that come into play? How does the heart beat? How is blood pressure maintained? What are some common cardiovascular diseases? We'll review those. So I said, in terms of what the cardiovascular system is, I said it's a system that includes the heart and the blood vessels. Job brings nutrients to cells and to help get rid of waste. It also helps to refresh the lungs and the kidneys and the intestine and the liver. So if you say refreshing the lungs with what? With oxygen, bringing in oxygen from the, and then also getting rid of carbon dioxide, refreshing the blood, bringing in nutrients, will help the kidneys to get rid of stuff that, to clean the body and intestines, to be able to move things, absorb material, and then take things to the liver, and we'll see how it comes to play for all of these things. Meanwhile, the lymphatic vessels help the system to collect excess fluid from around tissues and cells, and then to return, if it is excess, to bring it back into the cardiovascular system. They kind of balance each other. So these are the functions here of the four main ones: generate blood pressure, transport blood, exchange nutrients and waste at capillary locations, regulate blood flow as needed. So in terms of the transport function, so if the cardiovascular system helps to transport material, what kind of material does it transport? Oxygen, yes. Carbon dioxide, and other waste products that we do, and nutrients and hormones. All those are carried and distributed, either getting rid of carbon dioxide, bringing it to the lungs for it to be sent out, or oxygen to be brought in, taken from the lung areas, as I was mentioning, nutrients after digestion, hormones from their organs or their site of production to areas where they will need to be used. In terms of the protective function that it has cells, like white blood cells of the immune system, and helps to transport them to protect the body from infections. Regulatory functions, we talked about it. When you are hot, your blood vessels will dilate, bringing the heat from inside your body in the blood to the surfaces of your skin so that it can escape in sweat. After the sweat captures the heat, then it can evaporate. It also regulates what? pH balance, how acidic our blood is, and then also the electrolyte balance, what kind of ions we have, the sodium ions, the potassium ions, the various kinds of ions, to make sure that they are all in balance. You can click on this animation to watch more details about it. So the homeostatic, you can look at this and then at your spare time. We've talked about it. Meanwhile, the lymphatic system's job is what? To assist again the cardiovascular system. They work side by side. And then, what is that? Thank you. Thank you for letting me know. I was just going on and on and on. Let's see this here. So zoom back here. Thanks for pointing this out. Go back here. I hope you're seeing something this time. Excellent. Please support. Can you hear me now?
Yes.
Okay. So I was just mentioning that these are the functions of the cardiovascular system. I was just pointing out here, but you could have heard what I said without looking at the picture. But when I said the animation that talks about how our cardiovascular system does all of this stuff is located in this hyperlink right here. And you can also click here to see the same animation. Now, the lymphatic system, I said it works in parallel, actually supportive function of the cardiovascular system, in that when you have excess fluid in the surrounding extracellular fluid or something like that, it goes into this system, and then sometimes it will return into the blood. Then we have some arteries here, some lymphatic vessels here that flow into our blood vessels. So this is the path flow of blood within the cardiovascular system. It leaves from the heart. It is carried by some blood vessels called arteries, which are larger and thicker. The idea is, coming from the heart, the blood is under tremendous amount of pressure, and then under that pressure, you want the blood vessels not to explode, so the walls have to be thick and to be able to carry the blood under that pressure. And these blood vessels go as they branch out to various parts of the body, right to the surfaces of our skin, to the individual cells. They go from large, thick, to smaller branches, then to one cell size blood vessels, capillaries, which are where the cells are kind of distributed. And the cells they mesh. It's almost like they are surrounding cells, and the blood is not under any much pressure. It allows for gas exchange and everything to exchange in that situation. Now, when they have exchange of material within the capillary level between cells and the vessels, the blood vessels the capillary starts joining together to form venules, which are slightly larger than the veins, than the capillaries, and then venules will combine to form veins that are larger, and then will bring the blood back to the heart. That is the path. Give me 1 second here. Let me check that something here. I think this is an example of the capillaries. Give me 1 second. I'm going to put this out here. I'm going to stop sharing for a bit. Come in this direction here. Here. Give me 1 second. All right, let's go back here and see where we were. Now you see, when I was talking about the blood vessels, if you look at the arteries on the left compared to the veins on the right, not as thick. The inside here, they call the lumen of the artery, is slightly smaller. But what is also missing inside here is that it doesn't have valves. Look at the vein. They have valves here, which means that when blood goes through here, it cannot come back. So if the heart was from this area where my cursor is, pumping the blood, it will come through this artery here. The biggest one is called the aorta. And then the aorta now would branch into smaller arteries. And then the arteries will branch into arterioles, slightly smaller. And then here you have this level here called the capillary bed. The movement of blood from this direction is based on the pressure provided by the contraction of the heart. And the pressure starts decreasing as the surface area here, because it's spreading here into many branches, it starts reducing and then gets to become the lowest at the capillaries. And this allows, again, for material to exchange. Remember, we're talking about things moving in and out of either into the cell and out of the cell. So all those exchanges that are happening, whether it is diffusion of material, osmosis, or even active transport, all that kind of stuff happens here when the pressure is lowest. And the cells, all this is like birth. The cells are all birth in all of the small vessels. After it is done, if the cells had contributed like waste material into the blood vessels, DOO and carbon dioxide, it needs to be assembled now and taken back to the heart to be sent to the lungs, so that the lungs could now have the carbon dioxide removed and then rejuvenated with oxygen. But the veins start assembling here. The capillaries start assembling into venules, which are smaller, then the veins, and then the vena cava are very large veins. Some coming from the top region of your head region, some coming from the bottom part. So you will hear something like superior vena cava and inferior vena cava, all those then feeding into the heart. So what is it that brings blood, carries the blood, like from our legs to our heart? There is no generator on our feet or heart pumping. It is because we depend on our skeletal muscles and these valves. So our skeletal muscles would now contract and press on the walls of the blood vessels of the veins, which are not too thick, so that they are able to squeeze them a little bit. And then when they squeeze them, the blood will move from one compartment, because there's several. After this valve here, you go a little bit more. Again, you see another set of valves, and they're all like valves along the way, one-way valves. When you walk, you squeeze the blood. The skeletal muscles press on it, it squeezes up. All this action contributes and then moves the blood right back into the heart. So if you look here again, we're talking about cells. The outer layer of connective tissue, the inner layer of the that has the endothelium here, and then you have the layer of the smooth muscle cells right inside here. And if you see, this layer of smooth muscle cells here is thinner here than it is here. This is a better illustration again. This is like almost like a side by side. You see here, this is the wall of an artery. This is the one of a vein, larger and then much smaller and thicker. The one thing I would also like to point out in terms of this coloration is that they usually depict red if they're talking about blood and the organs or vessels that carry blood. When they show them red, it means that the blood it is carrying is rich in oxygen. When they're colored like blue like this, it means that the blood is rich in carbon dioxide. So this is how I was telling you about at the capillary level. You see how it just kind of forms a mesh, and these are individual cells. So we have those exchanges happening here in terms of material in and out of cells. And the material, sometimes if you see the material that is coming from the blood that is coming from the arteries to the arterioles, so because of that pressure, some of the material just even moves out, even material that the cell needs moves out. But as it gets further within the capillary, you would see that some of the material might even start diffusing back in. And then some from the cells. So you have cells here would put material into the extracellular space. That means because if the cells are not attached to one another, or even if they are attached, they're not like glued to one another. I said if you want to imagine some cells, they could be almost like cells have some distance between them and the next cell, and that distance is filled by, they call it extracellular fluid. So material exchange between the cell and as it gets into the blood vessel is material comes out of the cell into the extracellular fluid. From the extracellular fluid, it gets into the blood vessel and goes in the reverse direction as well. So at the end here, this is blood that was brought. As it is leaving to form, going back to the heart, you see now it's pouring oxygen. And you have waste that have come in, glucose that has gone out, carbon dioxide has come in, amino acids have gone out, useful things that the cell needs. Now, this is a schematic that illustrates the entire movement of blood. So you go here. It's very important for you to remember this. Let me see whether I have this situation here much better. I think there's a diagram that has a better illustration of this. If you're able to explain this to track here, when you look at your heart, when you are looking yourself in the mirror, your right side is your left side. So here, you would see that this is called the left part of the heart. This is the right part of the heart. The heart, you will see here, it moves from the heart. Blood moves from here through the aorta. It branches from here going towards our up head region area, and then some of it goes to our feet, our trunk area that is below the heart, and then it distributes it to all of these organs and assembles. They start assembling the blood back and coming in here and bringing it to the heart again. I said there are large veins that bring blood back to the heart. The ones from the bottom after the venules and the veins have merged is called the inferior vena cava. The one that comes from the heart, from the head region and above your chest area is called superior, like above, superior vena cava. Now, we have a system where blood is taken through the rest of the body, but we also have a situation where blood needs to leave from the heart and go to the lungs so that it could be refreshed, meaning get rid of carbon dioxide and take up oxygen from the lungs, and then be brought back to the lungs for renal to be sent again to the rest of the body. So we call of this one a systemic circuit, where it goes throughout the rest of the body, and then where it just goes from the heart to the lungs and back to the heart is called the pulmonary circuit. Again, the picture I was illustrating in terms of the differences between the arteries and the veins. If you look at the heart, on the other hand, the heart is about, it's a muscular organ. They usually refer to it as a myocardium. I say it is surrounded by a sac called the pericardium, as we mentioned earlier. It's about the size of our fist. And then when you cut it open, first of all, as a tissue itself, as an organ itself, it has blood vessels and take up waste from them and so forth. So even though it's pumping blood to the rest of the body, it's also got some blood vessels that are giving these muscles here nutrients and getting out their waste again. So if you cut this open, the best way to look at it is almost like a quadrant, like a square with 4 sections, 2 top sections and 2 bottom sections. But if it is a square, you would imagine that all the top portions at the bottom section should be the same, but that's not the case. So it does have 2 upper sections called the atria. This is the left atrium. Atria is both of them. Atrium is one. So the left atrium and the right atrium. The lower portions of them, you have the left ventricle and the right ventricle. Separating the top by this valve called semilunar valves, and they call them atrioventricular valves. Atrioventricular, this is a ventricular valve. Sometimes this one has 2 things that are holding it, connecting it to the tissue. They call it a bicuspid valve. Meanwhile, this one is also an atrioventricular valve. And you see now, these other 2 things connecting tissue that are holding it together, they are called, because there are 3, sometimes they call it tricuspid valve. And I said blood comes from the top section and then you see this one here, you see now, this is a blood vessel that is bringing blood here and bringing blood here. And then when it comes here, something's going on here. This chamber, this left side here, and actually, hold on 1 second here. This one here, the one thing that this picture doesn't show here is that separating the left side is totally separated from the right side. The things that is separating the left from the side is called the septum, a muscle that separates the left side from the right side. And the left side has blood that is oxygenated, that will eventually be sent, comes in here from these 2 here are blood vessels coming from the lungs to feed this left atrium, and the blood will move here to the left ventricle and then will be pushed out here through the aorta. Meanwhile, now here, this is the superior vena cava is bringing in deoxygenated blood that will come here and then move into the right ventricle and then will move out here to the lungs so that it can get refreshed, meaning that the lungs can remove the carbon dioxide and add oxygen to it. A better picture of the heart. And see here the coloration: red blood vessels, oxygen rich; blue painted blood vessels, referring to the veins that are poor in oxygen. And the one thing that I could also note here is that if you look at it, all the vessels, when blood is being squeezed out of the heart, most of it is deoxygenated blood. They only have one blood vessel that is carrying, one artery that is carrying oxygen, that is carrying deoxygenated blood. The rest of the arteries carry oxygen. The pulmonary artery, which is the one that is carrying blood away from the heart to the lungs. The only vein that is carrying oxygenated blood is the pulmonary vein that is coming from the lungs. Take note of that. Again, this is another picture, again, illustrating the same things as you can see, but with just different coloration and distinction for you to see. This is what I was pointing out to you earlier. That this one is the pulmonary circuit you could see moving from here. The systemic circuit is the one that goes all around. Look at what is happening here. This deoxygenated blood comes here. The lungs do what? The lungs send out the carbon dioxide leaves from here by diffusion, comes out into the lungs. Oxygen goes into the blood vessels, and the same thing goes. So the most important thing here is to kind of understand the path through which blood flows. I said you have the inferior and superior vena cava that are bringing blood into the right atrium, and then you have all of that. You should be able to track that and explain it to somebody. Again, this is another picture that shows you the same thing. Now, in terms of what makes the heart to beat, it's kind of a very interesting thing. And then you would also hear in terms of blood pressure. Really, what is pressure? The blood is the amount of force that the blood is exerting on the walls of the vessel. That's blood pressure, simply. So you can imagine that, as I was indicating, the vessels that are taking blood away from the heart should be thicker, and then also should, so because they are under a lot of the blood there is pushing harder to the wall, so they have a higher blood pressure compared to the ones that are bringing blood in. I think this is not supposed to play, but there's a version of this that you're able to see, and so you see when the heart beats, it contracts. When it contracts, it is pushing the blood out, and you would they call that systolic phase. As when you see how they measure blood pressure, you will do that, and then when it contracts and then it relaxes, when it relaxes, all this the atria. So when it contracts, the ventricles are squeezing, and then when it relaxes, the blood was in the atrium would flow into the ventricle that have set the blood that used to be here, sent it out. The one thing that I should also point out here is that if you look at the walls here of the left ventricle and those of the right ventricle, you will notice that these walls, the muscle walls of the right ventricle, are much thinner than those of the right ventricle. And why could that be? Because the pressure, the thicker the walls, the more pressure they will be able to squeeze the liquid that is inside here. And since on the right ventricle, the blood is only being sent to the lungs, which is not as thick, they are able to be efficient to squeeze the blood to leave. Then subsequently, if you look on the right side, on the left ventricle, where they have to squeeze blood to go through the rest of the body, the blood has to be under more pressure, so you should have more thicker walls to be able to squeeze them. And that's really what it is. So you see a situation here where structure is suitable in forming its function. Thicker walls, higher pressure, pushing blood to the rest of the body. Less thick walls, enough pressure to send blood short distance to the lungs. And then you also notice here that we're talking about semilunar valves. When the blood when the ventricles contract, the blood is forced out. But when the ventricles relax, it will look like the blood should come back, the one that was already in the arteries going out. It doesn't come back. Why? Thanks to this one-way valve, this valve here is called semilunar valves, that would prevent backflow of blood. So I talked about contraction, systolic, and then the diastolic relaxation. And then I said this circle of contraction and relaxation It happens about 70 times a minute. Does it happen exactly at 70? I say around average times, around 70 times a minute. So what is it that controls the heart, makes the heart to beat? There is something here called the sinoatrial node on the right atrium. On the right atrium. And then when it would send electrical signals out, action potentials, they will go now, they will radiate, and then they will come to this other one's group of tissue here called the atrioventricular. Innovate, like the signal will swarm the rest of the ventricle section here, in the Purkinje fibers they call them, and then it will stimulate. When that electrical signal is going, then it makes the whole thing contract. So these impulses travel between gaps, junctions at the intercalate. You might remember we're talking about connection between cells. So we said they have gap junctions. And the intercalated discs that are holding the cells together. So the signals are amplified and they radiate and make everything to happen together. It's not like one person contracts it before the other one does, but everything goes in sync. Now, I said, what controls heartbeat? And I said, external control. This is internal control of heartbeat. Meanwhile, we also have external control of heartbeat, our brain, and then we have hormones or chemicals that could also control the rate at which our heart beats. Talked about it. I see the thing here, comes from here, they amplify it, it goes to the rest of this body. So the way of based on understanding how the heart contracts and relaxes, you can actually graph the pressures of during this duration. And we do this all on something called the EKG or ECG electrocardiogram. And it will show you here what is happening when the heart is contracting, and then what is happening when it's relaxing, and then the recovery, and then how this thing goes. So the pattern, the chart that is produced from reading your heartbeat, like on an EKG or an ECG, they call them, could help people. And then we take our blood pressure. We have many ways that we do it now, and they tell you how to take it. We are not but the point for us to know is that when you're taking blood pressure, you take the one during the contraction phase, called the systolic one, and then we take it when the heart relaxes, which is the diastolic phase. And the average pressure for the systolic one is 120 for adults, and then for the one for the diastolic phase is about 80. So when you said average, so what happens when it's too high or too low? If this is about average, above this systolic of about 95, or less than 95, when it's less than, if your systolic is less than 95, we say you are hypotension, you have low blood pressure. And then when it is above here, and we don't want it to get too high. Because if it gets too high, it can even pop your blood vessels, right? And your blood vessels are pop at the wrong area is not a good thing, and it's not difficult to manage. So I say, what is important about blood flow? Again, we're talking about pressure. From here, the pressure is highest, and the surface area here is less. As the surface area increases here towards the capillaries, the pressure is lowest, and the pressure is least here, but the surface area here is highest, which makes sense. And I said, in terms of pushing blood out, a heart contraction enables that to happen. To bring blood back to the heart, skeletal muscles help, and the valves that they have, and then in the breathing and all of that. So you see here, all of these are veins taking blood back to the heart. We said that even though the heart is its own organ, it cells to need to need nutrients and get rid of waste. So you have blood vessels called coronary arteries, coronary veins, those that serve the heart muscle. I said there's something also called the hepatic portal system. I said this system brings blood from the digestive tract, rich in the nutrients that it has absorbed. It takes it to the liver. In the liver, it can take them there. If there's more glucose there, some of the glucose can be converted into glycogen. And then I said the liver synthesizes blood proteins, so you can take some of the amino acids and build some of the proteins that we need, and stores glucose as glycogen, as I mentioned. The liver also plays a role in also removing some, they say, purification role from some stuff that might have come from the digestive tract that is not good. So I said, finally, the blood will return to the heart through the inferior vena cava. So our liver is lower than the heart. So when they go, that vein that carries it and collects and brings the blood from the lower section, including from the liver, and brings it to the heart, is the inferior vena cava. Some disorders associated with blood vessels, we hear hypertension, high blood pressure. Sometimes you have a situation where plaque gets built up in the lumen of the blood vessel, atherosclerosis. Some of it could result in stroke, where your blood vessels are messed up in the brain, and then if there are controlled areas which control certain sections of your body, then they can get affected. Heart attacks, when your heart either something goes wrong and then it doesn't beat the way it's supposed to beat. Some aneurysms can have a situation where there is a blood vessel sagging and from ballooning, and then having problems in your brain or in some other areas, or your stomach or something. So I'm going to kind of stop here. Does anyone have any question? I'm going to stop sharing for a bit. Does anyone have any question? Anybody still there?
Yes.
Okay. For today's lab, we are supposed to do, let me go back to our canvas here. I'm having some cold symptoms, which I don't think is a bright idea to come to class for us to do the lab there. So today we'll do this lab virtually, and which is a simulation of what we're supposed to do in the lab today to look at how enzymes work. So we're going to look at this lab has to do with how enzymes work. We look at parameters that involve. First we'll look at what affects enzymatic activities. We're going to look at the effect of pH on enzymes, the effect of temperature on enzymes, and also the concentration of substrate. The lab is on is from the same lab manual in your virtual labs in McGraw-Hill Connect. There's exercises, virtual experiments that you will do when next week when we come to class. we will do this, repeat some of these ones so that you have the real hands-on experience doing it. But the benefit of us doing it, even though you're going to do this lab now virtually, is that you would have seen how the experiment is set up is very similar to what we're going to do in the lab itself, and then we'll be able to do that much faster. So week 3 enzymes work. So as the effects of temperature and enzyme activity, you've known how to manipulate to move things on these simulations, and you should be able to go through that easily. And then effects of pH, you also be able to do that as well. I think one has to do with enzyme. The one that has to do with enzyme concentration, we will do that when we do that lab manually. So you have these ones at the virtual labs that you're going to do for today's lab. We will not have in-person lab today. Does anyone have any question?
So, are we just going to do these by ourselves, or are we meeting online to do it?
Say that again.
So, are we going to meet online to do the lab, or are
No.
we just going to do it by
You're going to do
ourselves?
this by yourselves at your own
Okay.
time. Yeah.
Okay, thank you.
So no in-person lab today. And since we did not finish, the chapter that I want us to finish before we do the first exam is the cardiovascular system and blood. Since we did not finish this today, we cannot do the exam, we cannot do the midterm next week. Okay? So what will happen is that I will send you a schedule for our midterm, or a study guide. No, I will not send it. Go to finish reading this chapter on cardiovascular system. We will talk about blood, and then I think it doesn't make any difference. I will send you the study guide for the midterm, but the midterm is not going to be hard because I want us to have the lecture, answer your question. Okay, so the only thing you have to do this week is the lab and the quiz that is associated with chapter 4 that we talked about, tissues, the organization of the body into tissues, cells, tissues, and organ systems. Any question?
Professor, I have a quick question. Our lab was the one that was due yesterday, the one you're asking us to complete today, correct?
No. This lab I just I wanted you to just preview it, but was it due yesterday? Did I say the due date was yesterday?
It said there was a quiz this week saying quiz 2, and I think it was due yesterday. I don't know if that one's the one you're talking about, or you're gonna give us another quiz to do today.
Let me just check. What was the content of the quiz? Maybe I've messed up with what was covered in the quiz?
I think it was the first few chapters.
So the quiz of today that you're going to do this week has to cover the material that we talked about, tissues, and this first part of the cardiovascular system. Was that present in that quiz? Was this content present in the quiz? Okay. Yeah, so it will be separate from that one. I think the one you did yesterday, the last time, talked about like those proteins, those macromolecules, the thing that we did in chapter 2 and part of chapter 1. Some of the cell stuff, yeah. I just did the cell part here to just kind of review it again, but we had done that before, right? The only part that we did not do of the cell was talking about the cellular respiration part, how ATP is made. But in terms of the structure, talking about the various parts of the cell, I thought we talked about that, just generally what each part does. Did we talk about that last time?
I think we talked about it a little bit, but we didn't go too much into detail.
Did I give you the quiz that you took? Did you go too much into detail? Did I give you the quiz that covered that already?
In the quiz, there were some questions which had you label parts of the cell.
Mhm.
So Yeah.
Okay, so which was, and I told you that I wanted you to do to learn how to do is to name the part of it was to allow for you to be one of the objectives was for you to be able to know how to label those parts, right?
Yeah.
Yeah. So, and again, look at this like it's my way of forcing you. This quiz is my way of forcing you to review the material. So if I use that as an objective, then the quiz of this week would cover some of the stuff that I talked about, and then some of the stuff that I talked to what I talked today, and then and it will make you to read. If you want, I will also specify what the quiz will cover. What I cover today, like the introduction to most of what I talked about the cardiovascular system. It's not going to talk about blood. It's not going to talk about and the cardiovascular system part that is left. We didn't talk about the lymphatic system. That part we didn't mention that here. It's part of the cardiovascular system that we have not completed in this chapter that I stopped before. That I just kind of paused right here. So I will give you assignments only to the quiz will cover only what we have covered thus far. And I'm hoping that you can just review that so that when we finish it next week and then finish blood, then I will hopefully you should be able to, if you have identify any areas you do not understand, you can ask me, we can go over them for you to be able to understand. And then after that, then we can schedule whenever I want to have the midterm. The midterm is dictated when we do it is mostly dictated as of covering material, talking about it, asking you whether you've understood it, you telling me you don't have any more questions to ask about it, then I ask you to go do it because the goal is not to make you fail, is to make sure that you answer the questions. I answer your questions, especially in the areas that I've asked you to focus studies on the study guide. Then I ask you to go do the test. That's all. Is that fair?
I have a quick question.
Yes.
Midterm is on microwave or in person?
It might be remote. It might be just be like I think it'll be remote. But what I'm saying, it's just like an extended version of the quiz.
Okay, thank you.
As having been a student I hate to. I know that when somebody says it's an exam, you kind of freak out a little bit. But when I say it's a quiz, it looks like it's no big deal. Just look at the midterm as that we took the quizzes after 1 or 2 chapters. Now we're going to cover 6 chapters. And then I'm going to give you like an overview group big quiz on that section. And since the material is kind of a little bit large, so I will give you a study guide that points you into specific areas which I want you to know. That makes sense?
Yes, thank you.
Yeah. So I would stop here. It's about 4 o'clock. It's about not 4 o'clock. It's about it's 1 something 107.